So I'll go ahead and introduce Al Dubra. So Al Dubra, if you want to upload your slides. So he'll be speaking to us about adaptive optics, imaging of optic neuropathies.
Thank you, Shannon. I would like to start by congratulating Drs. Liao and Goldberg on their leadership in the creation of the center and thank them for the invitation to participate in this inaugural meeting. A few clinically available techniques have been discussed so far to study the effect or the impact of optic extrusion in patients. And today I'm going to talk to you about techniques that are still at the experimental stage, but hopefully will allow us to perform some new discoveries and learn about the disease in ways that we couldn't do before. So let me start by disclosing that I have been a consultant for a couple of companies, and none of them is relevant to the work today, and that we had some support from the National Eye Institute and RPV. So the learning objectives for today are to understand a little bit about what adaptive optics ophthalmoscopy can do for optic extrusion, and also review a couple of novel microscopic biomarkers that are applicable to these conditions. So if I can drive this, there you go. So the imaging, the current imaging paradigm for the retina in a clinical setting is that we have a relatively broad view of the retina, which is convenient to detect pathology. But in the case of optic extrusion, we have the advantage that we can actually target our attention or imaging just to a restricted area here in the optic disc, and also near the fovea where there is nerve fiber layer loss. But the traditional imaging paradigm is that we look at a large portion of the retina, and if we were to zoom in there, even if we have sufficient pixels, the images would be blurry. And not because the instruments are bad, but because the optics of our own human eye are imperfect. So if you could correct for those imperfections that are unique to every individual eye, and in fact also every retina location, you could start to see individual cells. And this really represents a major change in the way we study any eye disease, let alone optics extrusion. And in this case here, I am showing you the pixels of the eye that initiate the process of vision by absorbing light. These are the cone photoreceptors, and if you squint in the image on the right, you will see that there are smaller features in between them that would be the rod photoreceptors. So adaptive optics works by borrowing technology developed for astronomy that takes some of the light that comes back from the retina when we are imaging the retina and diverts it to a wavefront sensor that is very much like the ones used for refractive surgery in the eye clinic. And they take pictures with a camera of an array of spots that tell us what the optical imperfections of every individual eye are. And then we feed that information to some device that changes shape, in this case, a deformable mirror. And this allows us to create much higher resolutions than we could in the past. So this device effectively transforms our ability to look in the retina so that we are now looking through a microscope. And let me illustrate this again, the idea of the scale of the technique. Here on the right, on the left side, you get to see the typical view that you get in the retina. And this bright rectangle here shows you some of the light that we're using to take pictures with adaptive optics instrument. And that rectangle, if you zoom in, then you would get the images on the right, where you see much, much finer detail. And in this case, on the top right, you get to see these major blood vessels near the optic disc. And in the bottom, you see individual cells, again, the photoreceptors. So why do we think this is important for optic disc drusen? Because for almost every single eye condition and even systemic condition that we've looked at, we've made some discoveries. There is a wealth of microscopic features that cannot be detected in any other way that are not known even by pathologists that have been studying the retinas of donor eyes for decades. So we've discovered features that are punctate, that are associated to blood vessels, features that macroscopically they might look alike, like epiretinal membranes that have very different textures and might point us to many different disease mechanisms, and also have commonalities between different conditions that, at least in the current clinical understanding, are not related. And here I show on the left a number of conditions that we've imaged that have features, and each gray square here represents a type of feature that has been found in patients with these conditions. And the point here is to illustrate that there might be a lot more interaction between different conditions or common mechanisms that we currently think. And you might notice here that there is no optic disc drusen on the left because this has not been explored yet. So we're very excited about exploring this avenue. And one of the things that is very important to study in optic disc drusen is the blood flow, as Dr. Liao and Dr. Maus alluded earlier today. And one of the advantages of looking at the microscopic scale is that you could look at the flow without having to use any contrast agent on a very, very fine scale. So if you could see this video, I hope it shows on your screens, there are some distorted, elongated features flowing along the vessels here, and each of those corresponds to a single red blood cell. In fact, if you were to take that movie and average it or calculate a standard deviation like you would do in fluorescein angiography, then you would get a map of perfusion, which is of course very useful. But it's even more useful to be able to look at the structure of the capillaries even when they are not perfused, which is what we can see here on the right side. And also something that by looking at the microscopic scale, we could start doing in optic disc drusen is to study not only the flow, but also the structure. Are the blood vessel walls affected in any way? And is there something that we can see here, for example, that would be, that would precede changes in flow that might be already indicating an advanced stage of the condition? Then here, let me show you an example of the most extremely minute vascular accident that you can possibly detect. And this is in human retina. And you might see that there are 3 red blood cells here indicated by the arrows that get literally stuck for a few seconds. And with every heartbeat, they kind of move a little bit forward, eventually unclogging the blood vessel, the capillary, and then the movie plays again. So anyway, so this gives you an idea that you have the ultimate sensitivity in terms of vascular accidents. And then also another feature another part of the retina that we need to explore with these imaging modalities is the nerve fiber layer. So as of today, optical coherence tomography allows you to look at the overall thickness of the nerve fiber layer, but now we want to look at the intensity of the light reflected by the nerve fibers. And here you can see these striations that run diagonal from top right to the bottom left, and those corresponds to nerve fiber layer bundles. So these are very small image that is only about 150 microns across. And you can see that there are some bright dots that appear to change in brightness. And just to focus your attention in one of them, so let me zoom in one of them. And this is movie is playing on real time. So you can see that this feature becomes dim and then bright again a few seconds later. So we think that this is indicative of axonal transport that is thought to be affected in optic dystrophin. And if we were to take images every 7 minutes, which is a little bit longer than the movie that I played before, you would see these fairly dramatic changes in the intensity pattern that are not an artifactual speckle, but rather a real set of microscopic features that are changing. And we want to explore if these patterns change in optic dystrophin, and also if they change as the disease progresses. In addition to looking at the retina in the scale of seconds or minutes, we can also look maybe in the scale of hours. And this is, although not practical for clinical care, it might be informative in terms of disease mechanisms, because we have been able to show in other diseases, in this case, Best's vitelliform macular dystrophy, which is a progressive condition where the photoreceptors are affected and eventually die, where we can see this structure here highlighted by the arrow that, sorry, apologies for the incorrect labeling here. The two images are shown at time 0 and then 60 minutes later. And you can see that this structure seems to move within the retina. And even though it's only a few microns, it might be suggestive that we're looking at macrophages that would be cells that would appear in areas of cell loss and would be indicative of disease activity. Again, getting a little bit longer in terms of time scale, one of the challenges that we face with diseases that are slow progressing is how can we have sensitive measurements so that we can steer treatment, if it was available, faster. And here is an example of another neuropathy. In this case, we are talking about glaucoma, where we can see how there are some features that have been found in other optic neuropathies in addition to glaucoma. And there are inner nuclear layer microcysts. So these microcysts could change in the span of only a few weeks. And this is an example of how you could see like the boundary of the lesion kind of moving from the bottom of the image to the top of the image, where there are some of these microcysts present on the bottom left and very few on the top left here. And as the disease progresses, then we see that some of these features disappear and new ones appear at different locations. So these are actually telling us how the disease is actually moving spatially. And then in more longer term and in collaboration with Dr. Moss, in fact, she's the project leader here, we've been looking at multiple sclerosis patients that have shown some features that in portions of the retina that typically you would not think as associated to optic neuropathies, like the very center of the fovea, at the bottom of the foveal pit. And this is, here I'm showing you images from a normal control. And then if you were to look at a multiple sclerosis patient, you would see that there are all these cell-like structures in the foveal pit. And we don't know if these are present in other optic neuropathies or what the implications are. But we're hoping that we can use the same methods to learn about optic dystrophin. And then this is all the imaging that I've shown you so far is structural. But one of the things that we need to look at in the future is, can we tell not only the presence or the health of a cell by the structure, but can we see functional changes? Can we see changes in the chemistry of individual cells? And we're hoping to do so by using a widely used family of techniques in optics, in biomedical applications and industrial applications, that is spectroscopy. And the idea here is to exploit the different wavelengths of light so that we can learn about the chemistry of individual cells. And here we're showing you the surface of the nerve fiber layer, where you can see those bright reflective dots that I showed you before in black and white, but now in color. And these could potentially tell us about the health status of these, maybe in a snapshot, rather than having to wait for them to change. And
Well,
thank you
this is
for your
Shannon.
attention. I'll be happy to take questions.
Great. Thank you so much, Alf.